Hello and welcome to all my dear students. So, today we are going to start with the next lecture of the chapter Molecular Basis of Inheritance. So far you have understood the mechanism of replication. Now you have seen that from how DNA is copied. You have seen that the 100% of DNA is copied to produce the new daughter strands. I mean to say, both the parental DNA strands, they act as template, they undergo replication. Now, one of the step of central dogma, that is replication, is done. Today we are going to start with the second step of the process, which is called as transcription. Transcription means formation of RNA from DNA. Formation of RNA from DNA with the help of the enzyme DNA dependent RNA polymerase, because this time our DNA is going to act as a template, and DNA acting as a template, RNA has to be synthesized, has to be polymerized. Definitely, therefore the name of the enzyme will be DNA dependent RNA polymerase. So what is transcription? Transcription simply means formation of RNA from DNA with the help of enzyme DNA dependent RNA polymerase. Correct? Right? Now beta, unlike that of in replication, you have seen where both the strands of the DNA are acting as template, and on both the strands the replication process takes place to synthesize the new DNA. However, in case of RNA synthesis, in case of transcription, out of the two strands of the DNA, only one of the strand undergoes transcription. The strand which undergoes transcription is known as template or non-coding. Otherwise, the other strand with the polarity 5' to 3' is known as non-template or coding strand. First difference between replication and transcription. Second difference is, unlike that of DNA, whole DNA is copied. Whole parental DNA, as I told you, is copied to produce new DNA. But in case of transcription, a part of DNA segment undergoes transcription, and that part of DNA which undergoes transcription is known as transcription unit. So first I'll be writing the 2 points of difference, and then we will come to transcription unit. First point. Unlike that of replication, right? Unlike that of replication, comma, in transcription, only one of the DNA template, only in case of transcription, only one of the parent or one of the DNA strand is transcribed, which is known as template or non-coding strand. First point, right? That means suppose this is my DNA, correct? Polarity will be, the two strands will be anti-parallel to each other. If one of the strand is 3' to 5', then automatically the other strand will become 5' to 3'. Okay. Now synthesis always has to take place. Polymerization always takes place in 5' to 3'. That means the polarity of the template has to be in 3' to 5'. So now, if these are my DNA strand, correct? So strand with polarity 3' to 5' act as a DNA template. So that means this strand is going to act as a template for myself. Template strand is also known as non-coding. Now this strand will be read with the help of the enzyme DNA dependent RNA polymerase to synthesize a complementary RNA strand in 5' to 3' direction. Remember beta, RNA is always single stranded. Unlike that of DNA, RNA is always single stranded and synthesis but obviously has to take place in 5' to 3' direction. Now you will think that ma'am it is known as template. Okay, because enzyme RNA polymerase is reading this strand, so therefore this DNA strand is known as template. But why it is called as non-coding? The reason behind that why this 3' to 5' strand of DNA is known as non-coding, as later this RNA will be used to synthesize proteins. Correct? Okay. One thing beta more, this template DNA, the arrangement of nucleotide will be exactly complementary to the arrangement of nucleotides found in the RNA. I mean to say, if over here the arrangement is ATGC, ATGC, AAAAA, like for example. So on the complementary side over here, the nucleotides will be complementary to adenine is thymine, adenine, cytosine, guanine, thymine, adenine, cytosine, guanine, thymine, 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 thymine. This is going to be the arrangement of nucleotide on the non-template strand, which is also known as coding strand. Whereas the arrangement of nucleotide on template strand will be this. Now can you tell me what will be the arrangement of nucleotide on the RNA? It is going to be the same as that of non-template strand, simply thymine will be replaced by uracil. That means instead of A over here it will come U. T, A, C, G, U, A, C, G, U, 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 U. So can you say one thing, that the arrangement of nucleotide in non-template and in RNA synthesized is same, except thymine is replaced by uracil. Otherwise, the arrangement of nucleotide in non-template strand and RNA strand is same. Right? Both are complementary to the non-coding strand, also known as template strand. Now beta, it means that whatever protein RNA is going to be synthesized, whatever protein RNA is going to synthesize will actually be the proteins of the nucleotide arranged on non-template strand. So in short, you can say that the protein synthesized by RNA is dictated by the non-template strand, because the arrangement of nucleotide on this RNA is same as that of non-template strand. That is why non-template strand is known as coding strand. Coding means the strand which is utilized for protein synthesis. So basically non-template strand is same as that of RNA. Whatever protein RNA is going to synthesize is actually the same as that of non-template strand. I hope you have understood this point, that why non-template is coding and why template is non-coding. It all depends upon the arrangement of nucleotide. First point. So first point of difference you have seen between replication and transcription. Now moving towards the next point of difference. Now unlike that of DNA, or unlike that of replication, comma, during transcription, only a part of DNA is transcribed. Only a part of DNA is transcribed, called as transcription unit. So now you have to see that what is transcription unit. Correct? Okay. Now, suppose this is my DNA. Both the strands are anti-parallel. The template strand has polarity in 3' to 5', and non-template, also known as coding, has the polarity in 5' to 3'. Now, this whole DNA is not going to undergo translation. A part of the template strand will undergo transcription. Suppose this is the region. This part of DNA is going to undergo transcription. So the unit, the part of DNA which undergoes transcription is known as transcription unit. Correct? Now, the portion of the DNA beta, the portion of the DNA which is left before transcription unit, this is known as upstream region. Whereas the portion of DNA which is left later on over here, this is known as downstream region. Clear? Yes or no? Okay. So this is basically coding strand and this is your non-coding. Now beta, see, this transcription unit can further be divided into 3 parts. The portion of transcription unit which is present towards the 5' end of coding or 3' end of non-coding strand, it is known as promoter region. It is known as promoter region. So, first section of transcription unit is beta promoter. Promoter is present at the 5' end of coding or 3' end of non-coding. Usually, the polarity of transcription unit is decided by the coding strand. Correct? So, according to coding strand, write it. According to beta coding strand, comma, promoter is located at 5' end. Clear? Now, last towards the 3' end of coding or towards the 5' end of non-coding is present terminator region. And in between promoter and terminator is present your structural region. So transcription unit can be divided into 3 sections: promoter, structural and terminator. Promoter, again I am repeating, it is present at the 5' end of the coding or 3' end of the non-coding. Correct? Promoter region consists of some special sequences of nucleotide which allows the initiation of transcription. So this promoter consists of certain specific sequences. Certain sequences are present on the promoter region which are called as initiation region. So this initiation region is present in promoter region of the transcription unit, which helps in the correct initiation of transcription. So promoter, first of all, where it is located, I just hope you all have understood. Second, promoter region consists of special initiation nucleotide sequence so that transcription process can start from the right location. Correct? Okay. After coming from promoter, the second part is terminator. Let me just add a page first. Terminator beta, terminator, the name itself suggests it is the last segment of the transcription unit which marks the end of the process of transcription. So where it is located at the 3' end of coding or 5' end of non-coding strand. Now you know the polarity very clearly, very well. Second point, this region marks the end of the process, right? This region marks the end of the process of transcription. Understood, everyone? Now, in between promoter and terminator is present structural region. The structural region that flanks between promoter and terminator is that part of the template DNA which has to actually undergo transcription. So this structural is the region which flanks between promoter and terminator. So structural region is the region which flanks between promoter and terminator. I hope you have understood what is transcription unit. It is that part of DNA which undergoes transcription. It can be divided into 3 regions: promoter, transstructural and terminator. Promoter marks the beginning region of the transcription, whereas terminator marks the end of the process. For this whole thing, for whole transcription, enzyme RNA polymerase is required. So next we are going to talk about the enzyme. Now enzyme required beta for this process, I have already told you in the beginning, is the full name is beta DNA dependent RNA polymerase. Correct? Beta, in case of prokaryotes, only one RNA polymerase is required to synthesize all the types of RNA, tRNA, mRNA, rRNA. Only one RNA polymerase is there in prokaryotes that performs synthesis of every RNA. However, in case of eukaryotes, there are 3 types of RNA polymerase. RNA polymerase 1, polymerase 2 and polymerase 3 that helps in the synthesis of different types of RNA. So you are going to write in this case 1 for prokaryote, right? And 3 RNA polymerase for eukaryote. Clear? Okay, whatever be the number, but if I talk about the structure of this special enzyme, then beta this is a complex enzyme. Complex means that this enzyme is made up of 2 components. Each component has their own specific role to play during the transcription. The 2 components of RNA polymerase are sigma factor and the core enzyme. Right? Yes or no? Sigma factor and the core enzyme. Sigma factor along with core enzyme. Okay, this core enzyme beta, it is again made up of 4 more components. Each component of core enzyme has their own specific role to play. The core enzyme is made up of alpha, beta, beta dash and omega. So this is core and this is sigma. Both these components together, they form RNA polymerase enzyme. Clear? Both these components together, they make RNA polymerase enzyme. Clear? And this enzyme helps in the process of transcription of the transcription unit. It is present 1 for prokaryote and there are 3 different types of RNA polymerase for eukaryote. In case of eukaryote, every different polymerase has a role to synthesize specific RNA only. I'll tell you later on that which RNA polymerase in eukaryote synthesizes which sets of RNA. Okay. Now, before I start with the process of transcription, one more thing I want to share it with you. I hope so far you have understood everything, whatever I have written on board. Transcription क्या होता है? Transcription is a process where DNA is copied to form RNA. Correct? Second, for this process, a complex enzyme is required called as DNA-dependent RNA polymerase. Correct? Third role of transcription is in understanding the transcription unit. That whole DNA does not undergo transcription. Only a segment of DNA undergoes transcription called as transcription unit, which is divided into 3 segments as initiator or promoter, structural, and then terminator. Right? I hope you all have understood whatever I have written on the screen. If yes, then let's proceed further with the next concept, and then we will jump into the process of transcription. Beta. Now, see, in case of eukaryotes, beta, transcription is monocystronic. In case of eukaryotes, transcription is monocystronic. Whereas in case of prokaryotes, beta, transcription is polycystronic. Now, what is this monocystronic and polycystronic? So first of all, let me define the term cistron for you. See, mono means one, poly means many. Cistron is that segment of DNA. It is that segment of gene which is active in protein synthesis. So write this note point somewhere in your notes. I am writing over here. You also please write this somewhere in your notes that cistron represents that unit or region of DNA/gene which is beta responsible for protein synthesis. Correct? Now, monocystronic means in a gene there is only one active segment to produce protein. Polycystronic means that in a gene there's a DNA where you can obtain many active regions for synthesizing protein. Clear? Poly means many, cistron means active region. Okay, so what happens in case of eukaryote beta? Check this. Suppose this is eukaryotic DNA. Clear? Polarity reverse. Now, suppose this is the transcription unit. Now, in one segment, in one stretch of DNA, there will be only one transcription unit with one promoter, one whole structural, and one whole terminator. Now, this structural will synthesize only one polypeptide chain at a time. So it is monocystronic. But in case of prokaryote, suppose this is DNA. Strands are anti-parallel. Correct? Now, in one segment of DNA, there will be many active regions called as many transcription unit. Promoter, structural, terminator. Again, a promoter may come. Structural, terminator. So from one segment of DNA, you obtain many polypeptide chain. Now this is one polypeptide chain, this is another polypeptide chain from one segment of DNA. So it is basically many, more than one polypeptide chain are obtained. So this is polycystronic. right? So I have to remember that eukaryotes are monocystronic, whereas prokaryotes are polycystronic. Apart from it, beta, in case of eukaryote, being monocystronic, like at a stretch only one polypeptide is obtained, but their structural gene, their DNA, their template DNA is also having some non-coding, some interrupting sequences called interons, which are absent in case of prokaryote. So you have to write this note point somewhere, that in eukaryotic DNA is not continuous. Whole DNA of eukaryote at a stretch cannot produce protein. So eukaryotic DNA is interrupted by presence of non-coding segments. Non-coding segments called as interons, and these interons, beta, they are absent in the DNA of prokaryote. So eukaryotes DNA, they are rich in interon. Okay, the coding sequences are known as exon, but non-coding sequences are known as interon. Non-coding means that part of DNA which cannot produce protein. But however, this non-coding regions, they are absent in case of prokaryote. Okay, so these are the details, minute details, which you should know before we start with the process of transcription. So exons are basically coding sequences. In case of prokaryotic DNA, lacks interons. This is the complete information now I have written on the board, so that you all can copy in your notes as well. Nothing should be missed out from your notes. So please try to copy everything, whatever is written on the screen. Chalo. Now we have to start the process of transcription. But again, before that, you know that there are different types of RNA. Mainly there are three types of RNA: mRNA, rRNA, and tRNA. In case of prokaryotes, all these three are synthesized by one enzyme, that is RNA polymerase. But in case of eukaryotes, these three types of RNA are synthesized by three different types of RNA polymerase. So first, let's get an information about the three types of RNA. Okay, there are three types of RNA are mRNA, rRNA, tRNA. Now, majority is 80% of RNA is rRNA, 15% is tRNA, and 5% out of the total is mRNA. So which is most abundant in the cytoplasm? rRNA. Which is least? mRNA. Correct? Coming to the second. Now if I talk about the size, mRNA being in least quantity, it is still the longest mRNA, which is acting as a messenger for protein synthesis. rRNA are in mid, but tRNA are the smallest ones. Clear? Now, talking about the roles. If I talk about beta mRNA, see, roles if I talk about of individual RNAs, first I will talk about mRNA. M stands for beta messenger. Right? Messenger RNA means the RNA, it is a full 5' to 3' single stranded RNA which carries the information for protein synthesis. So mRNA carries the information for protein synthesis. When I'll teach you translation now, then there you will see the exact role of every RNA in protein synthesis. Right now we are just doing the theoretical part, but once I'll teach you the concept of translation, then there you will see that how every RNA is playing the role in translation in protein synthesis. Next is tRNA. Beta, T stands for transfer. It is also called as adapter RNA. Why? Because its function is, suppose this is mRNA. Now, mRNA is having some set of nucleotides which need to be read so that it can be converted into protein. Correct? Now, to produce protein from these nucleotide, amino acid should come and bind with them. So, to join amino acid on the messenger RNA, tRNA are utilized. So, basically, tRNA helps in attaching amino acid on the mRNA for protein synthesis. On the mRNA for protein synthesis. Right? Now, third and most important, which is the most abundant one, 80% rRNA. R stands for beta, ribosomal RNA. They have many roles. They have catalytic role. They can be used as catalyst. You study in zoology ribozymes. Ribozymes are RNA based enzyme. Which RNA? rRNA. rRNA like 23S and 28S in prokaryote and eukaryote respectively, they help in making the enzymes. So rRNA have catalytic role. Apart from this, they also make ribosomes. The units of ribosome, 70S, 30S, 50S, 80S, 60S, 40S, all these units are made up of different types of rRNA. So they help in making ribosomes, right? Thus, they help in translation. Understood? Now, individually, I have told you the functions of every RNA, but actually, more you will understand once we will start with the translation process. There you will see that how individual RNA is helping in the process of protein synthesis. Right now, just the theoretical part has been done. Practical you will see when I'll teach you translation. Chalo. So now background is done. Now you have to just become the warrior and come and stand in front of the field. That is, now you're going to study about the process of transcription. Now, this process of transcription beta can be first divided into 3 steps. Very simple process. Initiation, elongation and termination. Now, in case of beta prokaryote, this process is taking place in the cytoplasm. But in case of eukaryote, this process is taking place in the nucleus where replication took place. Now in prokaryote, there is no nucleus, there are no other cell organelles. Every metabolic activity in prokaryote is directly taking place in the cytoplasm. So replication also took place in cytoplasm. Transcription is also taking place in cytoplasm. But if I talk about eukaryotes, then you know eukaryotes, they have well-defined nucleus, cell organelles are found. So the work of replication and transcription is done in nucleus itself. Okay, chalo. Now let's start. Now you know the location, you know the enzyme, you know everything about transcription unit. So let's start the process of initiation. Initiation. Okay. Now suppose this is my double-stranded DNA. See. Now both the strands of DNA are helically coiled. Polarity will be reverse. Oh yeah. Okay. Now this whole DNA is obviously not going to act as a template. Man lo, only this much part of DNA is acting as the template. So this is known as transcription unit. Okay. Now before the region of Yes, this region which is present prior to transcription unit, which is upstream, is having some specific recognition sequences, or you can say like this. Let me draw it once again so that diagram is more clear. Suppose this is my double-stranded DNA. Now right now these double-stranded DNA beta hydrogen bonds are prevailing between them. So they are helically coiled and they are very much in contact with each other. Like this. Okay. Now beta, what will happen? A part of the DNA will undergo transcription. Suppose this is the region. This is that transcription unit which is going to undergo transcription. Here is the promoter region. This is suppose structural region. And at last is the termination region from where the process is going to end. Now beta, in the promoter region are present some specific sequences. This is 3' to 5'. So on the 3' end of the promoter region, there are some specific sequences called as recognition sequence. Right? These, whenever transcription has to start, first of all, sigma factor. See, right now RNA polymerase, which is of 2 component, when transcription is not taking place, both the components are separate from each other. Right? When transcription has to start, first sigma factor comes and recognizes the recognition sequence in the promoter region and comes and get bind to it. So it comes and get bind to the recognition sequence. So initiation means correct binding of sigma factor in the promoter region. As soon as sigma factor binds to the correct sequence, it calls the core enzyme that okay, now I have done my job. So now core enzyme will come and bind with the sigma factor to complete the set of enzyme. So what is initiation? Initiation means binding of, first step of initiation means binding of sigma factor to the recognition sequence. Beta, present in the promoter region. Okay. Now, once it is done, second step, then core enzyme binds to it. Ab as soon as beta core enzyme attaches, now core enzyme also have helicase property. Once core enzyme binds, RNA polymerase is complete. Now this core enzyme has the capacity of helicase. Now as soon as the correct binding takes place, as soon as RNA polymerase is complete, now core enzyme starts reading the template strand and starts adding nucleoside triphosphate complementary to the parental strand and starts synthesizing the RNA. And how? Because right now the DNA is double-stranded, but it has the helicase property. So now it is going to start the unwinding. Now RNA polymerase will keep moving. Once initiation is done, once core enzyme is happy, sigma factor then detaches. Then comes the next step, which is elongation. Once elongation begins, means once the proper transcription begins, sigma factor detaches. Now only core enzyme is left. Core enzyme will help in elongation. How? As it has helicase property, so it will keep on unwinding the DNA duplex, will make the DNA single-stranded, will read the template strand, will add the nucleoside triphosphate complementary to the template strand, and yes, will continue the process. So core enzyme will help in elongation as it has helicase property, comma, thus first makes the DNA duplex into DNA into single strand form, comma, then reads the template and adds nucleoside triphosphate and then adds nucleoside triphosphate complementary to template strand. And then adds complementary to template strand, right? Like this. See, if suppose here this was the promoter region. Now this is structural region, and at last there is terminator region. Now promoter region was having some sequences where the work is done. Now sigma factor got detached. Now core enzyme is moving ahead. It is unwinding the DNA duplex. Right here is somewhere the core enzyme. Now this is the template strand. This is the template strand. Now this core enzyme is reading the nucleotides of the template strand and adding the complementary nucleoside triphosphate and making the RNA chain. And this process is going to continue till the time core enzyme will enter into the termination region. So the last step of elongation is beta termination. So what happens in termination? Once your core enzyme reaches to the terminator point of the transcription unit, then rho factor is another factor like sigma factor. Sigma factor was responsible for correct initiation. Rho factors are responsible for correct ending of the transcription. Rho factor comes and bind with the core enzyme. It stops the further activity of core enzyme, separates the core enzyme from the template strand, separates the RNA, and thus the process comes to an halt. So what is termination? With the help of rho factor, process of transcription ends. How? Rho factor binds with core enzyme and thus recognizes the end sequences to stop the process. To stop the process. Clear? So these are the three major steps of transcription: initiation, sigma factor; elongation, core enzyme; termination, rho factor. These three are important things which can be asked to you in the form of questions. Now whatever be the method, your transcription is completely done. Okay. Now beta, in case of prokaryote, as soon as transcription is completed, even sometimes even when transcription is continuing, translation is also taking place simultaneously. But in case of eukaryote, transcription and translation are not coupled. So note point. Beta, in case of prokaryotes, both the process, both transcription and translation are coupled. Coupled means they're going hand in hand. Once transcription start, translation, like transcription is going on and पीछे से translation भी होता जा रहा है. So in case of eukaryotes, sorry, in case of prokaryotes, both the process are coupled. However, this is not the case in eukaryote. In eukaryote, transcription and translation are two different events taking place in two different compartments of the cell in two different times. Are coupled, however, not in eukaryote. Now, what is the reason behind it? Now, question comes in mind that kya karan hoga? What is the reason that why in case of eukaryotes, these two processes, transcription and translation, are not coupled? What can be the reason? So let's check out the three reasons behind that why in case of transcription the process is not coupled. Okay, so let me tell you, I think I have not mentioned over here, no issues, let me tell you. In case of beta eukaryote, first reason is transcription is taking place in nucleus, but translation, mRNA is required for translation, which is found in the cytoplasm. So translation has to take place in cytoplasm. Now, when two processes are taking place in two different compartments, definitely they cannot be coupled. Unlike that of in prokaryote, both were taking place in the cytoplasm. No compartment has to be changed, so they got coupled. But in case of eukaryote, since beta transcription takes place in where? Nucleus, and translation takes place in cytoplasm, thus they cannot be coupled. How can they be coupled? Now first, once mRNA will be obtained, then mRNA has to move to cytoplasm, and then only their tRNAs vagera is present, so that translation can start. So first reason. Second reason, now beta, in case of eukaryote, DNA is rich in interons. You know, non-coding sequence I just told you. So these yellow colored are beta interons, non-coding sequence. Now when this DNA is used for transcription, then RNA is synthesized. This RNA polymerase also transcribes the interons. So these are interons. In between the interons, there are coding sequences which are called as exons. Right? Now this RNA which is obtained after transcription in eukaryote is rich in interons and exon both. Such RNA is known as heterogeneous RNA. This is known as hnRNA. Heterogeneous, the RNA which is not uniform. It is having both coding and non-coding sequences. So now this hnRNA beta, it cannot undergo translation. First, interons need to be removed. Till the time interons will not be removed, the process cannot start. So this is the second important reason that why in eukaryote both the processes are not coupled. First reason is compartment is different. So till the time RNA will not come out of the nucleus, process cannot start. Second, RNA which is obtained in eukaryote is heterogeneous. Why? It is rich in both interon and exon. So therefore, first interons need to be removed. A common RNA, a homogeneous RNA has to be synthesized. Then only translation can start. So these are certain reasons that why in eukaryote, translation and transcription are not coupled. But in case of prokaryote, since compartment has not to be changed, since there are no interons, therefore, in case of prokaryote, both the processes got coupled. Understood the reason? Okay, if yes, then let's proceed I told you one thing, that in case of eukaryote, there are 3 different RNA polymerase required for transcription. But in case of prokaryote, only one RNA polymerase can do the same process. So just check it out, which RNA polymerase is required for the synthesis of which RNA. Read it. There are 3 types of transcripting enzymes, that is RNA polymerase in the nucleus in addition to RNA polymerase found in the new organelles. There's a clear-cut division of labor. RNA polymerase is responsible for the synthesis of 5.8S, 18S, 28S rRNA. RNA polymerase 2 is responsible for the synthesis of the RNA that I was talking about, heterogeneous. And RNA polymerase 3 is responsible for tRNA, snRNA, 5S RNA. Clear? So you can see in case of eukaryote, the synthesis of RNA is divided between 3 types of RNA polymerase. I hope you all have understood till here. The types of RNA polymerase, the process of transcription, monocystronic, polycystronic. Yes? If yes, then I'm going to keep my lecture till here. Further post-transcriptional modification in eukaryotic RNA we are going to do in our next class. Not today. For today, till here only. Simple, short and crisp. So today we covered everything related to transcription. I hope you all have understood. If yes, then let's proceed further and check out some questions. So these questions are just for you to attempt so that you can develop a mind that have you understood the concept of transcription or not. So with this, I would like to say simple thank you, and yes, keep watching the lectures, keep yourself updated with the information given to you on daily basis on this amazing channel of PW English, which is absolutely made free for all those students who are really willing to do something great in your lives. Thank you for joining me. Bye bye everyone. Take care.